Hi, I'm John Flannery. I'm visiting from California. I'm a professor at the University of California, and I work on inherited retinal degenerations therapies. I think it's critical for genetic testing because the phrase we like to use is no gene therapy. So we have to design a gene therapy on the basis of knowing what the genes are. And so the only way we know what the genes are is from genotyping the patients. And then we go from there, and in the laboratory what we do is we try to figure out what those genes do, and we try to make small animal models that have the same gene defect as the patients, and we use those animals to test potential therapies. Currently, all the therapies that are in the clinical trial, of which there's about 12, they are each all specific for particular genes. And so the way the current ones work is that you identify by testing the patient what the exact gene is, and then your therapy is actually to replace that defective gene with a mutation with a normal copy of it. So what's critical about that for genetic testing is that first, in order to know what gene to put back, you have to get it from the patient, and then you design the therapy around that. But secondly, I think what's also important is that if you have gene X, a therapy for some other patient will not only not work for you, it actually could make you worse. So it's important that you know exactly what the defect is in the patient and that the therapy is exactly the right one for that patient. So you can be not appropriate for a particular therapy because you have a different defective gene, but it's very critical to know exactly what your gene is to design the therapy. So the more genes that we know, the more therapies we can develop. It's also important for dealing not only with researchers, but for dealing with companies, for they know how many patients have a particular disease gene so that they can run a trial. And what we mean by running a trial is get a collection of patients that have the same gene defect and then test them. And so unless you know what the gene defects are, you can't get a collection of people from different backgrounds that have the same defect. And then also there's, as I said, there's therapies that are what we call gene replacement, where we identify what your gene is and give you back a healthy, non-mutated copy of that. But there's other therapies that are not gene specific. So depending on what you have, there may be a therapy that exactly replaces your defective gene, but there may be another therapy that works for many different patients, or maybe a subset of the patients, and that you may be appropriate for that. So we can't make those decisions unless we know the genotype of the patients, and the more patients that are genotyped, the better. And then another part of that equation is that as we get databases and collections of all the patients in a particular country, in a particular city, and around the world, then companies make decisions about, is there 100,000 people with this defect? Is there 20,000 people? Are they mostly in Ireland? Are they mostly in the States? And things like that, so that they can design what are the best targets and how prevalent the diseases are. So that only comes from getting large numbers of patients genotyped.